So now it's streaming live. Swapna ma'am, your chat box is appearing. You can just close it. I mean, okay. Yeah.
Okay, good morning, everyone. Shall I start? So today's topic is actually, good morning. Today's topic is actually introduction to heredity. So today briefly we'll get introduced to Mendel, who actually got the first few insights into heredity, and then we can go ahead next week. So let's start. So this session has been possible because of financial assistance from Indian Oil Corporation Limited. And I have shown you two pictures here. Which one do you see? The one on the left or the one on the right bottom? Which one is the more common picture?
On the right bottom.
So basically the point I was making was that whenever, anywhere, whether it is plants or animals, whatever we see around us, we always see that the new generation is always similar to its parents, right? So this is our general observation since time immemorial. And let me ask you another question, that new generation is always similar to its parent, but is it really completely identical? Yes.
813196 इसमें भेजा हुआ है.
Dori, you are audible to us, so you were talking to someone else, no? So ma'am is asking a question. I will request the children to answer, or even others can answer. It's not as if only the children can answer. We have now Dori and Kushal, but the Pondicherry group, you can also answer if you want. If nobody responds, the session does not advance well, because it's the whole session is designed with questions. So please answer. Yeah. So you can either speak or type.
Type.
Ma'am, you can cross this. There's a cross on top, and just cross this. Yes.
So the question that humans have been wondering about is, what is the reason of, or what is the mechanism, why the newer generation is always similar to its parents, right? Now, the next question is, Is the new generation exactly the same as previous one? Now we have shown you two conditions here. The one on the left you can see, and the one on the right, they are different. So sometimes we see that they are identical, sometimes we see that they are really not identical, right? So then in case of humans, you might say that that is specific to animals. So here we show you two sets of siblings. And you can see that although coming from the same parents, you can see the similarity, but you can also see that they are not really identical. Okay. Does anybody want to identify the names of these? Just a GK question, just to get you going.
The leftmost one.
Yes, Geeta, last name. So many Geeta. Geeta and Babita Fogart sisters. And the one on the bottom, maybe most of you are too young to know. Okay. Raj Kapoor, Shammi Kapoor, and Shashi Kapoor. Very good. Kushal. So now why humans have been wondering about this, not just because just a curiosity, but humans have been trying to breed plants and animals which are much more useful to them rather than what is found in the nature for thousands of years. So we have been doing this by which we classically call it as plant breeding or animal breeding. So we have been doing this for thousands of years, but it has never worked very well. Why it has not worked very well? Simply because we didn't understand what was really happening. So what was seen by people who were doing breeding for thousands of generations, that they never got consistent results. If they were, let's say, crossing to get a better yield or a better tasting fruit, it was not that they would always get what they wanted. What they would get sometimes would be something totally new, totally unexpected, probably something that is not desirable. And sometimes some characters which were important or necessary either completely disappeared or got modified. So you can see that breeding in case of dogs, we have hundreds of different varieties of dogs, and we keep getting newer and newer varieties. So, and in animal breeding, things were never under our control simply because we never really understood how characters are transferred from parent to the new offspring. Okay, so this was true in case of plants as well as animals. So right from Aristotle, Hippocrates, everyone has been wondering about how features are passed from one generation to the other. But now we know a lot about it. Now we are really in a position that we can really totally control which character is transferred or how to modify a particular character. Okay, so that is what we are going to understand in these couple of sessions as to how did we figure this out. So this whole process actually starts with a person called Gregor Mendel, who is also called as father of genetics. And he is considered father of genetics, not just because he was the first one to do so. People have been studying this for a number of years, but Mendel succeeded in a way that no one else did. And we will try to understand today why did he succeed where so many others failed. So he was the first person who got a clear understanding or clear insight into how different features are inherited, or why are they skipped, or why do they disappear. Okay, so let's first understand what this person, what he did. So Mendel was actually so ahead of his time. He was basically a priest who was interested in working in plant hybridization. And he was so ahead of his time that what he worked, this is his own handwriting that you can see, a page from his own notebook, that he was so ahead of his time that when he published his results, nobody really understood or appreciated what he was trying to say. And he actually got recognition 30 years after his death. Okay, so let's first see why he succeeded where no one else did. So he basically worked in his monastery's garden and what he did, his system was pea plants, and he basically consistently worked and analyzed thousands of pea plants every year. Okay, so this is basically what he worked on. Now, why peas? That is the question. So one reason why his experiments worked, one reason was because he chose a plant which was most suited for this experiment. Now let's see why. Okay, so he decided to study garden pea, that is the matarka plant. So why peas? So this is now the most important feature why pea works here is the structure of the pea flower. Okay, so here you can see that there are 3 pictures of pea flower, and you can see that this has a very unique structure, and the stamens and pistils, that is the male and female reproductive organs, are right here inside. So the structure is much different than the regular jasmine or sunflower or rose flower that we see. Now, what is so unique about stamens and pistils are hidden here. So basically what happens is this flower is designed for self-pollination, right? So pollination occurs even before the flower is completely open. So if you want to see the structure, this is the sepal, this is the petal. These 1, 2, 3, and 4 are the stamens or the male reproductive organs where pollen grains are produced. And this here is the stigma. This is the female reproductive organ. And these are the ovules where ultimately this, after fertilization, this will become the seeds or the peas. So this is how the pea flower is designed. So the petals remain closed. So the pollination generally that you see is self-pollination. Now, why this is important? Simply because you can always be sure of the parents of these seeds. Parent is only coming from this flower and not from outside. So you can really control who the parents are. This is the first feature.
Ma'am,
Now,
let's let's ask if this point is clear. I mean, self-pollination and that it's helpful. I mean, it is a desirable feature in this experiment. Firstly, Dori, Kushal, and Khushi, like the pollination bit and the self-pollination bit, do you understand? And also Madhvan, Reshma, and Kyle?
Yes,
I mean, the previous part, is it clear? Dori and Kushal, Khushi, is it clear?
Yes, ma'am.
Yes, great.
So is self-pollination very common? No, self-pollination is not very common in nature, but it is found in some plants, and pea is one of them. Okay. So now, does pea plant only allow self-pollination? No, you can also do cross-pollination. What do I mean by cross-pollination? Cross-pollination means that the fertilization is happening between pollens and the ovules in a flower. Okay, so if the pollens come from a different plant, then you call it as a cross-pollination. So the fertilization that is happening, the male and female gametes are coming from different plants. This is called as a cross-pollination. When they are coming from the same plant or even same flower, this is self-pollination. So using pea gives you an option of either doing self-pollination or doing cross-pollination. So this is very, very important. Now, second reason why peas worked for him is pea plants have a very short life cycle. So they grow and mature and produce seeds within a single season. So he was living and working in Austria, which has a very severe winter. So he was able to grow a complete cycle of pea plant life cycle. He could complete it in a single season. So this has a short life cycle. And another very important reason why pea worked for him is because pea plants show number of different features which have only 2 varieties. Okay. For example, here you can see that there are white flowers. So pea flowers, if you see, they either come as purple colored or white colored. The pea plants are either tall or they are short. So many of the features of a pea plant, you can only see two versions of it. And this became much more cleaner or easier for him to study. Okay. So there was nothing in between or these kind of features. So let's see some of these features. So you can have either purple flower or you can have a white flower. You can either have a tall plant or you can have a short plant. So if you look at humans, actually, if you compare height of humans, you see a variety from very short to very tall and everything in between. But in case of pea, it's either tall or short. There is nothing in between. So similarly, you can see round seeds, which are most commonly seen, or you can see wrinkled seeds. Only two versions of the shape. So like this, he basically chose 7 characters that he studied, which are plant height, flower color, flower position, seed shape, seed color, green or yellow, pod color, green or yellow, and pod shape, whether it is flat or wrinkled. Okay. So these are the 7. So these features that he studied are the technical word that we will use for this is a character. Okay. Now, and what you see, for example, plant height. So height is the character and the appearance is either tall or short. So tall and short, we will call it as a trait. Okay. So character and trait. So there are 7 characters and there are 2 traits for each. So 14 traits that he studied. So let's see, each character of a pea plant exists in 2 forms, which are called as traits in genetics. So for example, in case of height character, the 2 different traits or forms that you see is either the plant will be tall or the plant will be short. Is this clear? Okay, so let's go ahead. So let's see what experiments he did. Okay, now he had a very good mathematical training and he was mathematically oriented. So one thing he did was he had a very large sample size and he counted everything. He kept very good records and that's why he was able to analyze them. So let's see how he did the experiments. Now another feature that that sets apart his work from everybody else's is he isolated something called as a true breeding pea plants. So first 2 years of his decade-long work he spent in isolating true breeding or pure varieties. What do I mean by pure varieties? I'll just show you in 1 minute. So just reminding you that we are going to use 14 different traits, 2 traits at a time, and 7 different characters. So let's see what is a true breeding species. So what is the meaning of a true breeding pea plant? True breeding pea plant means he checked that all the seeds that he had isolated from a tall pea plant, if he germinates them, grows them, gets the flower, gets the seeds back again. So every time, all the seeds from a tall plant are going to give rise to only tall plant. There is no short plant coming from these seeds. So then he collected. So these seeds, he called it as true breeding tall. Similarly, he checked and rechecked a short pea plant that was always giving only short pea plants. Okay. So for 2 years, he did this for all 7 characters. He isolated 14 groups of seeds which were pure breeding. That means if he took any of the seeds from the tall seed collection, it would always give a tall plant. If he had a white flower collected seeds, then if he planted it, it would always give only white flowers. No purple colored flower will come in that. Okay. Is this clear? This is called as a true breeding. That is, that character or that trait is pure. There is no variation. So tall give rise to only tall children and tall grandchildren and tall great grandchildren. Okay, so he did this. Now another thing that I want you to remember is if he is going to take tall plants and short plants, these plants are only different in their height in all other respects, they are identical. Okay, so just remember this. So he had his 2 breeding varieties, and now he decided that what happens if he does a cross pollination, that he makes a hybrid from a 2 contrasting varieties, like purple flowered plant crossed with a white flowered plant, or a tall flowered plant crossed with short flower. So this is called as a cross breeding simply because we use cross pollination. Okay, so what this means is you are making a hybrid plant because the 2 parents are very different and they are basically having contrasting features. So here in this picture, we show you a white flower plant and a purple flower plant. So let's see how this is done. Now, cross pollination is not as easy. Okay. So what is done here is, first step is you need.
Ma'am, just take questions if there are any questions or doubts. I mean, on whatever you have said so far. Friends, I mean, what ma'am has said about true breeding and like what he was doing so far, is that very clear? I mean, specific characteristics and like each characteristic.
The plants.
Dori, though 2 devices must be interfering. So either you'll have to like off the mic of one of them, switch off, or write in the chat box. So Dori has asked, how do we get true breeding plants?
So you get, in the sense, you, let's see, Dori, this is a tall pea plant, right? So from the seed, this is grown, given flowers. So this is a tall pea plant. I collect the seeds from, so I collect all the peas that I get from this plant. Okay. And I plant it. Okay. So what will happen? Let's say if I collected 100 seeds from here, I planted all 100 seeds. So I would get 100 plants. And I really see that all 100 plants I got from this were all giving rise to tall plants. So this tall plant seeds only gave rise to tall plants, 100%. That is all of them. There was no short plant that he could get from this. So in this case,
which basically means that if there was any short plant here, he would throw it out. I mean, he would throw that seed. He will not include that seed. So as he would go through successive generations, he would remove the short ones, keep only the tall ones, so that finally only the tall, truly tall remain. Right?
So his collection was pure in that sense, that his tall varieties always gave rise to tall plants, no short plant in that. You understand, Dori? Okay. So he has now true breeding. Everybody else also understood?
Yes, ma'am. Understood.
Okay. So let's understand how to do cross-pollination. Now, as I showed you earlier, that normally in pea plant, what happens? The fertilization happens even before the flower opens. Okay. So if you want to do cross-pollination, let's see, then what should you do? You have to do something so that this flower does not self-pollinate itself. Correct? So what do we do is, suppose, let's see, this is the plant where I want to use the female, that is so if I want to keep this as a female, I should remove the stamens from this flower before they mature and before the pollen grains are released. So this is the way I can prevent self-pollination in this parent. How do I prevent self-pollination? Is simply I remove the stamens with a forceps and scissors before they mature and release the pollen, so that self-pollination is prevented. Is this step clear?
Yes.
Okay. So let's say this is the flower I want to use as a female parent. First step I would do is remove the stamens in their immature form. So this becomes a female flower because no stamens, no pollen grains. Okay. So this is how I prepare the flower. And now to do pollination, what I do is whichever, let's say I am crossing white flower with a purple flower plant. So this in purple flower plant, I have removed the stamen. So there is no pollen grains here. So I need to have pollen grains for fertilization. So I bring the pollen grains from a white flower with using simply a soft paintbrush. Okay. So using a paintbrush, you simply take some in the brush and apply it on the stigma. So now you have cross-pollinated from pollens from white flower is been transferred onto the stigma of the purple flower, which is a female flower simply because we have removed the stems. Is this clear? So one parent, you make it a female parent by removing stamens. So no pollens and no self-pollination. And then you bring in pollen grains from the second parent that you desire to use, simply using a paintbrush and apply it on the stigma so that pollination happens and eventually pea seeds will be formed. Is this clear how cross-pollination is done? If you do not do this, then only self-pollination will happen. Okay. So for cross-pollination, this intervention is needed.
So you so is my voice clear?
Repeat.
Is my voice clear?
Yeah.
Okay. So in the purple flower, if you remove the male part and take the female part of the white flower and then cross pollinate, or we take the male part of the white flower.
You tell
and
me which one
purple.
will you take? See, you have removed the male part from this flower.
No.
You have the
If we
female.
do the opposite,
You
if
can
we
do.
take,
He did the opposite also.
so
Okay,
the
but
result
let's.
would be same.
Yeah, the result was always the same. Okay, so every time you read in textbooks, anytime they show you one cross, but he did the opposite cross. That is the male-female version was reversed. That is called as a reciprocal cross. He did it for all 7 and he
Okay.
always got the same results.
Okay.
Okay. Is it clear, everyone?
Yeah, any other question? Like Pondicherry group? Any
No, ma'am.
other doubt or question?
Doubt.
Okay. So the first cross he did was he took purple colored flowers and crossed them with white colored flowers. Now, when Mendel started working, the mechanism of inheritance or heredity was understood to be a blending heredity. What do I mean by blending? It meant that you would get average result. For example, if I cross purple flowers with white color, I would get light purple colored flowers. So the features of the parents are being mixed or blended and you get a average or medium kind of appearance. So if I tall and short should give me medium height plants. So this was the accepted or popular theory at that time. That it's called as a blending inheritance. That is, you get the feature which is average of the 2 parents. So when he did his cross, he was also kind of expecting a pale purple flower. But what did he get? When he did the cross pollination between purple flowered and white flowered, what the seeds, so what he does is he does the cross pollination, collects the seeds, plants them, gets the plants and looks for what is the color of the flowers. So in contrast to the prevalent theory, which was a blending theory, he actually saw that when he cross pollinated purple and white flowers, he got all purple flowered plants. So he got a very surprising result. Okay. So first thing he did was he, as she asked also, he did a reverse cross. So that is, if this was the female parent, he did another cross where the white flower was the female parent and pollens came from purple plant. Again, same result. Then he did the similar cross with tall plants, short plants, again, same result. So he did it for all 7 characters, and every time he did cross-pollination and the plants that he got. Now, these plants, that is the offspring plants, are called as, in genetics, it is called as a F1 generation or the first generation. So this is parent generation, also called as P generation, and then you have F1 generation, that is the first generation hybrid. They all showed only single character, no mixing, and one character always disappeared. So in this case, the white flower character disappeared. Only the purple character showed. If he did tall and short, all plants were tall and shortness disappeared. Okay. So again, this is the same case that has I was telling you about, that when he crossed tall and short, he expected medium-sized children, but all new plants turned out to be tall plants. So this was really, really surprising. So he wanted to see what really happened. So this one character, whether it is white flower or whether it is short height, this character seems to have disappeared in the next generation. So this is happening every time.
Dori has raised a question, Swapna ma'am.
Yes, Dori. It didn't matter, even if you reverse the genders, the result was always the same. Okay. It didn't matter whether the purple was the female or purple flower was the male. The result of cross-pollination between purple and white was always purple flowers. And this was true for, so all 7 generations he crossed and he got similar results. That is, first generation hybrid is only showing the features of a single parent. Now, this was really very surprising. So, what is happening? Where is the other character going? Okay. What do you think? Where is the other character going? There are only 3 options that can happen. Does anyone know? Anyone wants to guess what is the... So he basically wanted to see whether... Yeah, go ahead.
It was hidden.
No, I'm
Because it's
asking
the another character is
possibilities.
dominant. The one character is dominant, that's why the plant.
But that he figured out after he did the next part. If you were where he is, what would you guess? This is what you are telling me is the result of his experiments ahead, right? This is not what he knew then. This is what we know now.
That may be the trait, one of the traits overpowered.
Dominant.
So the, yeah, what is... So one possibility was that the other trait was kind of eliminated by this. So purple color simply eliminated the white color trait. This was one possibility. Second possibility was that it was hiding it. Okay, not allowing it to express. So he wanted to see what is really happening, whether it is eliminated, whether it is masked, or it is totally destroyed. So what he did simply was that he collected the seeds from this first generation hybrid and he simply allowed them to germinate. So he got the plants. So first generation F1 seeds have been planted and the plants were allowed to grow, flower and give seeds. So this generation, so this is the first generation plant that he had. So he allowed the seeds to grow and checked what is now the feature in the second generation, which we call it as F2 generation. Okay. So we are starting with the seeds of the first generation hybrid. We are germinating them, getting the plants and collecting the seeds. And now sowing these seeds, which are going to be the second generation hybrid. And what was the result? So the result was, now when he collected the seeds and planted and checked the second generation, he got the disappeared character back again. Okay, so the character that didn't show up in the first generation showed itself in the second generation, which showed that it was not destroyed or eliminated by the other trait, but it was simply hiding. It was hiding, or it was masked, or it was covered by the other trait. Was there a fixed ratio? He counted the seeds and he saw that in the second generation, he always got 3 times one trait, and the disappeared trait showed up as in the 3:1 ratio. Okay, so this he got as far as plant height goes. This same result he got whether he did tall, short, and so on. So did you understand what is happening here? So this here we are simply showing you a visual of in his garden how he would have seen 3 times where the short trees are orange color shown here and the blue squares are representing the tall plants. So this is how his field would look like. So 75% of the plants showed the tall character and 25% of the plants showed the hidden character, that was the short character. And he got similar results. So this is when he decided to call this character which shows itself in the first generation as the dominant character and the one that hides as the recessive character. Clear?
Yeah, ma'am.
So, Swapna ma'am, you want me to do the matchstick activity?
Yes. Or do you want to do the complete activity next time, or we can do it now?
No, as you say, I mean, if you want to, but you have to talk about genes first, no?
Yeah.
Or no?
Yeah.
So first you talk about genes.
So let's review this again. What we saw in Mendel's cross-pollination experiment, or Mendel's crosses for short, is you start with a P generation where you have true breeding plants showing 2 different contrasting traits. Then when you cross-pollinate, get the seeds. This is called as the F1 generation, first hybrid generation. This shows only one of the traits and the other traits remain hidden. And when you take the seeds from this plant, allow them to grow and produce seeds, what you see is now you see 75% of the plants show the one character and 25% of the plant show the hidden character. Okay, so the ratio remains 3:1. So this is his first set of results. This allowed him to come up with explanation as to why this is happening. Okay, so he basically saw that this is a very universal finding that he got. For all 7 characters, he got similar result. So he had to now sit and figure out how he can explain the result. So he came up, and imagine the greatness of Mendel is that time barely it was understood what is a cell or what is inside a cell. Nothing really was known at that time. So even in spite of the lack of understanding about living organisms, he was able to make a leap and actually come up with an explanation which has really stood the test of time. So let's see. So this is what we've already discussed, that the shortness character, let's say, is absent in all F1 plants. So it could have disappeared, or it could have got destroyed. It was permanently changed by the tallness character, or it was hidden or masked by the tall character. So because this character appeared in the second generation, we know that this possibility was correct. It was neither changed, it was neither destroyed, or it was not made to disappear by the dominant character. So this was the first. So what did he infer? Let's see. So one of the biggest leaps that he made was this, that he proposed that every trait is actually controlled by a factor. Okay, and this factor is what is passed from parent to an offspring in an unchanged form. There is no change, because you can see that even if the shortness disappeared, it reappeared without any change. So when these factors are transferring between parent to offspring, there is no change that is happening, and they are being passed. So every trait is being passed from parent to the offspring through some factor. So this proposal of his was actually surprising, because before that, people did not think that there was a particulate factor. Okay, the blending theory was like an exchange of fluids. So this was a very surprising thing that he proposed, that a trait is actually controlled by a factor. So this trait, so let's say purple flowerness or purple color or white color, both are controlled by two different traits. They are passed from parent to offspring. Today, we call them genes. Okay, so Mendel called it as heritable factors. Now we know what they are, we know how they work, and this is the modern name that has been accepted is for factors is genes. So from here after, we'll use the word gene and not factor. Okay, so every trait is controlled by a particular gene, and this gene remains hidden in all the organisms. Okay, so this is his first proposal. Then further, he suggested that every plant, for that matter, every organism, must have a pair of genes for each character. Okay, so a parent has a pair of genes for every character, and so one gene is coming from mother and one gene is coming from a father. So every organism has a pair of genes, and this pair, when you have more than one, we call them alleles. Okay, so more than one gene, when it is present for the same trait, we call it, for same character, we call it a allele. So let's say, yes. So either you can say both alleles in the same organism can either be same or different. Let's see this in an image. So for example, when we started with a true breeding tall plant, which always gave rise to tall plants, both the alleles or both the genes in this parent must have been for tall trait. And when you similarly have a short variety, it was a true breeding variety, so always had short plants. That means both the genes in this short variety must have been the same, that is, they are for the shortness of character. And when we crossed tall with short, first generation hybrid that we got, it must have got one allele from the mother and one allele from the male parent, giving you a hybrid in which both the alleles are different. One is for tall character, one is for short character. And the standard convention in genetics is generally the dominant character is denoted by a capital letter, and the recessive character, which disappears in the first generation, is normally denoted by the smaller alphabet. Is this clear? Okay, the same thing in this case. It's, if you say, it's as far as flower color is the character, the trait is either purple color or white color. So purple color trait is controlled by, let's say, capital P gene, and white color is controlled by small p gene. And in case of height, capital T means tall trait, and small t means short character, similar for everything.
So, Swapna ma'am, I think now we can show the matchstick activity.
Okay.
So, who's sharing? You are sharing the PPT?
Yes. Sure.
So, you'll have to stop sharing. Then I will show it through my mobile.
Yeah.
So, I will. So, so you can, friends, you can hear me properly?
Yes, ma'am.
You will have to
And,
spotlight or something, ma'am.
Yes. So here's so what we are trying to do is see this will be useful for classroom. So that's why we invented this, that deliberately we are showing a tall plant and a short plant using matchsticks, and I mean taking a tall and a short matchstick. So initially he started with true breeding. True breeding means actually he ensured that he has the gene for that particular characteristic only. So let me make it, suppose, black. So this means the tallness gene is suppose black. So this plant, the tall plant, the true breeding one, which he started with, would have only the tallness gene. So genes are hidden inside. Nobody can see them, but that's what he guessed, or that's what the theory says. So here's a true breeding tall plant which has like tallness gene, and because it has only the tallness gene, it will, if you allow it to self-pollinate, it will always give rise to tall plants. So similarly for short ones, so the shortness gene is suppose red. So this plant has the shortness gene only. I mean, if this was big T, this is small t, and we have like, wait, just a sec. So since every cell has 2 copies of each gene, so this is the situation that the tall plant has only tallness gene, short plant has only shortness gene, and now they are now crossed, right? So for making the first generation, what he tried doing was crossing these two, which means like the sex cells of this one actually cross with sex cells of this one. So this will, whether it gives pollen or whether it gives ovum, the sex cell will only be black and black. So I mean, I will put, so sex cell has a single gene. This like maybe children know or they don't, I don't know, but sex cells, which are also called gametes, so which means like either it's the ovum or ovule in plants. I mean, it's the ovum or the pollen. So suppose this one is pollen, I will just write that. So this one is pollen and this one is ovule. So whether it's pollen or whether it's ovule, it will have only the tallness gene, single tallness gene. So black and black. And the same with this one. This one will only have the shortness gene. So whether it's pollen or whether it's ovule or ovum, I mean, that word is also used. So this will only have like red only. So now all this is hidden inside, but like we are kind of imagining, and that's why we are using these bindis, because it becomes easy to imagine. So dominant genes only and recessive genes only, and then they cross. So if you see that, suppose pollen from here combines with ovule from here. ovule or ovum, whatever. So this one combines with this. So the result is one black, one red. So the result is one black, one red. Now this ovule combines with this pollen, again one black, one red. Then so this pollen with ovule, ovule with pollen. What is the other combination possible? This, right?
Pollen with pollen.
The pollen will not combine with pollen. Basically, I mean, this one combines with, whether this combines with ovule or Swapnam, I should get 4 combinations, no?
It will
Anshumana
all-
ma'am,
Huh.
Yeah, go ahead, Nitin.
no, one plant gives only one gamete, right? I mean, that's the whole point of this, isn't it? So what he
It
was
gives
doing.
It
it
gives
gives
only one
pollen
type of
as well
gamete.
as ovules. See,
Okay, right.
yeah. So that's why I put in pollens and ovules. So if pollen from here combines with ovules from here, so you get this. If you and if ovules from here combine with pollen from here, you get this. Basically, you get only tall plants. I mean, okay, it doesn't matter. I mean, I don't have to show 4. It just means that if this is the combination of genes, the resulting plant would be a tall plant because the tallness gene, the black one, will dominate, right? So basically, this is the first generation. So first generation always have whatever combination is produced. The first generation has has both genes, but it will appear tall always because the tallness gene will dominate, right? So the point is actually that, I mean, so I'll have to show it here that way so that it becomes very clear. I'm taking a little time taking out these bindis because I'm like right now I don't have somebody else to help me with bindis. So, but it's okay. Taking a little time. Yeah. So basically now anything results from crossing of the first of these true breeds will always be this kind of mixture of genes, both genes present, tall and short both present, but the resulting plant is tall only simply because there is the black one is dominating, right? Which means like the dominant gene is there, so recessive one doesn't show. So this is the F1. So this is F1. Yeah. All plants of F1 are tall. Now, in the second generation, now the F1s are allowed to cross with each other. Actually, self-pollinate. Basically, self-pollinate. They are allowed to not cross. They are allowed to self-pollinate. So what happens if now this one gives, once again, so it gives. So let me just take one from F1, one plant from F1, which self-pollinates. So it gives now sex cells. So it can make sex cells like this. It can make pollen of this kind and also ovum of this kind. I mean, it's capable of making pollen and ovum, both of this kind, ovum or ovule, right? And also the same plant, okay, let me put it here. The same plant can also make red pollens and black pollens. Black pollens and black ovums, which means sorry. I mean, I must be using red. So these plants, there are many of them, but any plant can have pollen having the dominant gene, or ovum having the dominant gene, or pollen having the recessive gene, or ovum having the recessive gene. Now it crosses with itself, so it makes which means like there are all combinations made. So one combination will be this pollen combining with this ovum. Result, this pollen.
Black, red.
Yeah, you are asking something. Yeah. Dori, some question, or Dori or somebody else who spoke?
Someone spoke.
Is there a question?
No, no question. I just saying the answer.
Okay. So see, this pollen can combine with this ovum. So I'm making this and this combination. So I get this result. Then I can combine this. No, actually, I should have.
Ma'am, I think it
No,
will be easier if you make it into a Punnett square. So one side with pollen and one side with ovum.
Wait. I mean, what is the mistake I'm making? Let me just
It-
see.
No,
This
just
I've done.
show. Now you do from the black pollen to black ovum.
Black pollen to black ovum. Yes. This crossing will also be possible. No, I mean that's what. Yeah. Because I put, what I made the mistake was I should have written both pollens here and both ovums here. Then crossing would have been easier. So, but it's okay. I mean, so this and this pollen will also cross with this ovum because it's self-pollinating. So the result would be this, and then this pollen, this combination I made. Now this pollen and this ovum cross. So then we get one red and one black again. So actually only 4 combinations are possible. So all the 4 combinations will have, like, out of the 4. So this is this with this. No, sorry.
Yeah,
This
black
is
with black,
this with this.
black
This
with
with
red,
this.
and red.
I have
Now
made
red
this
with red.
and yes, this red with red. Yes, correct. Yeah, I should have written them reverse. Then would have been simpler. So actually, I should make one red pollen and one black pollen. Keep them here, and one red ovum here and one black ovum here, and keep them here. So then it becomes kind of very easy. So this is the combination in F2 generation. So all 4 possibilities are here. So we can now see that this one will give a tall plant because the tallness gene is dominating. This, where is my matchbox? Here. This is both tall. So again, a tall plant. One tall, one short, again a tall plant. And both short, so a short plant. So here, because the one combination gets both recessive genes, so the short variety reappears in 1/4 of the plant. So short, tall, and one short. So it's like you can do it easily with or show it easily with bindis and matchstick. The idea is that well, you can represent the tallness with a tall matchstick, and or like full matchstick, and shortness with a short matchstick. That's the advantage. And genes are actually not visible. So this exercise actually makes it clear how each plant is making its sex cells, its gametes, and then they are combining to give the next generation. So I got a little confused, but I hope you understand. Right?
Yes,
So I'll
ma'am.
close. Yeah. And any question? When you're doing this one, both
No,
columns
ma'am.
keep here and both ovums keep here. Then it will be simple. Okay. So I will stop.
So, from the matchstick activity, now it must have become clear that we have pair of genes for each character. Each trait is controlled by one gene, and then totally, if there are two forms, we can have three combinations. That is, both the alleles can either be the same or both alleles can be different. Okay. So in case of parents, we had capital P in case of purple flower and small p in a white flower. And when the hybrid was formed, we had one capital P from here, one short p from here. So the hybrid had capital P, small p. Okay. So next postulate, next proposal that he made was this. That is, when two different alleles are present together in one organism, then generally one allele dominates over the other. Okay. So when two different alleles are present, for example, in the first hybrid generation, you see effect of only one gene because one gene dominates over the other gene. So this is also called as law of dominance. So this is his second proposal. So this is just in case you had trouble with for understanding fertilization, just a quick primer. Every parent produces gamete cells or sex cells for reproduction. So one flower generally has both male and female gametes produced. So pollen brings in the male gamete and ovules bring in the female gamete. So ovule is present right at the bottom center of a plant, and pollens have to travel from the stamens onto the stigma and then actually travel all the way into the ovule for fertilization, which gives rise to seeds. Now, dominant and recessive alleles are represented in uppercase and lowercase. So, for a purple flower plant would always have capital P, and tall plant would have capital T. So, parent plant gives its genes to gametes. So this is just a recap of the matchstick activity. This is the tall plant forming pollens which are capital T or capital P, and ovum also are the same, identical. And your female parent, which is the short plant, would only have gametes which are only capital T. So when they bring together, when the fertilization happens, the offspring gets one gene from the father or the male parent and one gene from the female parent. So we just did the matchstick activity. So this is your first tall versus short. First hybrid is all tall. And then F1 when it was selfed, the gametes that are formed with the capital T and small t. So two types of gametes, capital T, small t, capital T, small t. So fertilization can be between these two, between these two, between these two, and between these two. giving you your 3 tall is to 1 short. So these are 3 postulates of Mendel, that genetic characters, that is heritable characters, are controlled by genes that exist in pair in individual organism. Second is when these 2 different alleles that are responsible for a single character, are different. In that case, one allele is dominant over the other. This is also known as the law of dominance. And third postulate is during the formation of gametes, the pair of alleles separate or segregate randomly so that each gamete receives only one allele with equal likelihood. This is also known as law of segregation. Okay. So today we have understood 2 laws of Mendel, and the next law, which is the law of independent assortment, we'll cover next time. So now in 200 years, where have we progressed? We have progressed from understanding of garden peas to understanding that every organism has cell. In the cell, if we look, they have chromosomes, and the chromosomes actually contain DNA. And this DNA, which is really what makes the gene. Okay. Now, Mendel's principles, when they were rediscovered 30 years later, they were different scientists actually tested them out with different organisms from peas, beans, sunflower, cotton, wheat, barley, tomatoes, maize, and many other ornamental plants, as well as with these animal species. And every time it was found that Mendel's laws were universal, and thus Mendel was called as the father of genetics. Now, garden peas is very easy. Actually, it's very easy to, relatively easy to work with plants. Still possible to work with animals, but how to work out inheritance of human beings? Because in case of human beings, you cannot do controlled experiments. So how does anyone know how do we do experiments in case of humans? We can't do crosses in humans, right? So what can we do? How do we figure out genetics of human beings? Anyone knows? Yes or no? Madhavan, but yes, I am not, that is there, that is universal in case of all organisms. One copy of a gene is inherited from mother and one is received from the father. But how do we study inheritance of characters in case of humans? That is what I was asking. Because you can't simply do crosses like that. So does anyone know how we study human inheritance?
Don't know, ma'am.
Okay. So actually, in case of human beings, what is done is you have to follow family history. Okay. So you have to, anytime completely different or a slightly unusual trait is seen, you have to follow the family history and do something called as a pedigree analysis. So you have to go back and figure out whether the parents, grandparents, whether we can figure out how this character was being passed, and you have to also follow the next few generations. Okay, so in the case of humans, this is mostly done by family history analysis, which is technically called as pedigree analysis. But for simple understanding, for simple discussion, or in as a classroom activity, what are the features in case of humans which are very easy to study? So I've shown you one. So these are all very simple properties one can study in groups anywhere. So there is this ability called as a tongue rolling activity. So either you can roll your tongue like this or you cannot. So these are simple features in a sense that they have only two versions. Either you can roll or you cannot roll. Okay. So these are much more simpler features rather than eye color, skin color, hair color, so on, and or height. In case of humans, these are very complex characters to study. So these are simpler characters to study. So you can go ahead and try to see if all of you, whether you are a roller or a non-roller. Okay. So how many of you are rollers and how many of I am a roller. I can roll my tongue. Can all of you roll your tongue?
Yes, ma'am. I also can roll
All?
my.
No. Okay. Generally, okay, that we have at least Preeti who is not roller. So you can actually go on Google and see which is the dominant. I'm not revealing whether which is the dominant and which is the recessive character. Similarly, another feature is this, very simple. Whether the earlobes are attached or not attached. That these are called as free earlobes and these are called as attached earlobes. Okay, so in your family, go ahead and see. This is a nice activity, especially for the students who are here to follow it up in your own family, especially if you have 3 generations going, that you can actually see whether, and you can try to predict whether which one do you think is the dominant feature. Okay, so if you are sitting in a group, you can obviously look at each other and figure out whether you have attached earlobes or free earlobes, or after this session, please go and look up the mirror and figure out whether you have attached or free earlobes. So this is another, that there are many such features which are shown here. So there is a bent little finger, cleft in the chin, then dimples, either you can have dimples or not have it. Hand clasping, so this is how you clasp your fingers, and tongue rolling. These are some very, very simple features that one can see. This is a widow's peak. This is called as, so you have this kind of a hairline. Can you see this triangular peak, which is not there here? So this hairline, which is like this, is called as a widow's peak. And as you can see, it's a dominant. So is the free earlobe. So these are the simple human characters that one can see. Now, human height, we saw that there is nothing like tall or short. So you can have anytime, I think between around 3 feet all the way up to 8 feet. So this is, human height doesn't have 2 forms. It has multiple forms. This is a very complex character because now we understand that this is not controlled by a single gene. It is controlled by many different genes. Skin color is the same thing. It's a range of skin colors from very fair to very dark. And again, skin color is determined by multiple genes together. So it's not a simple 3:1 ratio that we will get in skin color. Okay. And so these are called as polygenic inheritances. So this is just a map of the world and showing skin color. As you can see, closer to the equator, you have darker skin color. And as you go towards either poles, that you see the skin is fairer because it's actually how it works So more of an adaptation, what allows you to survive in high sunlight. So how many genes in, so different organisms have different number of genes, and just a number, this doesn't really mean anything. Garden pea has 41,000 genes, whereas humans have less. Surprising that humans only have about 31,000 genes. Cress, this is a small plant, has 26,000 genes. Nematode has 18,000 genes. Fruit fly, which you see as the small insects which come and sit on fruits or especially bananas, they have 13,000 genes. Yeast, which is responsible for giving us our daily bread, has only 6,000 genes, and bacteria have even less. Okay, so this is just an estimate, just a general knowledge fact. There are many frogs also which have much more genes than humans. So actually there is no correlation as there is this organism is better or worse or complex or simple based on genes alone. It's not a simple correlation. Now, do genes of all organisms follow Mendel's laws? Any comment here? What do you think? What is your opinion? Do all genes of all organisms follow Mendel's laws? What is your prediction, or what is? We called it Mendel's laws, right? So it's a law. So what do you think? Genes of all organisms must be following Mendel's law. No, Madhavan says no. So any idea which, anything extra to say, Madhavan? Which gene or any example that you know of? So basically, many genes do. Most asked example in many of our webinars is this. That's why we have added. So this is in case of human blood groups. So we talked about that whenever you have two different alleles, one dominates over the other. But in many case of many genes, you don't see this complete dominance. So in case of blood group, you have three alleles actually. Either is blood group A, B, or O. So there are three alleles A, B, and O. So you can have either AA obviously is blood group A. When both the alleles are B, it's obviously blood group B. But what happens is when you have one gene for A and one gene for O, in case of A, O is basically a recessive allele. So whenever you have A and O, you only see A blood group. When you have B and O, B dominates over O, so you see only B blood group. But what happens when one parent gives you A allele and one parent gives you B allele? In that case, your allele composition is AB. So in case of when you have AB, that is you have two different alleles, in this case, there is no domination between A and B. So you see both, expression of both. So blood group is AB. So this kind of example where you see both the alleles show their effect together, this is called as a codominance. So there are many such examples. So this is called as a codominance, and we will discuss the principle of independent assortment. So basically, independent assortment is what happens when you do a cross between plants which are different in two characters. You try to follow two characters at the same time. What happens, that we will see in the next session. If you have any doubts or queries, please go ahead and ask.
So no question from the Pondicherry group. So the children here, do you want to ask anything? So no questions from children also. So shall I then